Happy Friday, living soil nerds, and welcome back to Bioactive Live Q&A, where we're trying to connect with you guys, get some questions from you. I'm Mark from MI Beneficials. I'm here today with Brian from Rubber Ducky Isopods co-hosting, Kevin from OK Calyx and OKCalyxShop.com co-hosting, and we've got an amazing guest here. She's going to take us on a deep dive under the microscope to take a look at some samples of some compost that Kevin sent, some IMO that Kevin sent, and to try to help us to understand what it is that's going on in IMO and good compost. And so, without any further ado, I'd like to welcome Organic Alexa to the show. Welcome, Alexa.
Hi, thank you so much for having me. Super excited to look at these samples under the microscope. They are amazing, really biologically active. So I have a lot I want to say about them. And
Awesome.
it's a cool opportunity. I've wanted to do live microscopy online for a while. So this is a cool way to try it out and get started doing it. So thank you.
Hell yeah. Yeah, we've had actually several people ask us about this, and you're the first person that comes to mind.
Cool.
Um, I know Alexa through the community here in Michigan. She's a Michigan girl. I know professionally you work at Haifa Grown. We've had Ross on the show talking about Haifa Grown, which is a commercial no-till grow in a commercial setting. And we'll talk a little bit about that with Alexa. But beyond that, I just wanted to also touch on the fact that she's a passionate soil savant. We were talking about this earlier, and you can just get the feeling from someone like Alexa that she just gets passion from sharing this knowledge. So this is a special opportunity for everybody. We really hope you guys drop questions in so that we can keep this interactive. And again, the best question, I put it on Instagram, but the best question tonight gets a free biodiversity pack shipped, no cost to you. We'll pick it at the end. So keep that in mind as motivation. Um, so how does the kind of your microscopic view and your passion for microbiology, how does that help you at Hypha Grown in a commercial cannabis setting?
Wow. I mean, it's been instrumental, like really without it, I I don't know. So we started out I did not know anything really about microbiology. I knew it was important, but that's really all I knew about it as far as like leveraging it and growing it in compost and compost teas, I really had no knowledge. And so we started the grow, and um, I've been growing cannabis for 10 years. And first 5 years, it was, in synthetic systems. So when we moved into living soil, working or doing HIFA, it was like a huge change. It's, I've always been interested in organic farming, and I that's what I wanted to do and got the opportunity to do it with Haifa, which was amazing. And so when we started out, I quickly learned that it's not as easy as it looks. We had problems with pests and disease for the first few harvests, and I was like, okay, what's going on, doing lots of research. I found Dr. Elaine Ingham's course, and I was like, okay, I got to take this. And I did. And immediately I started applying these techniques to the grow, making compost teas and extracts. With that compost, and immediately saw the difference. All the pests and disease problems went away. Lush green growth, and it's um been that took me to like, okay, wow, these microbes are amazing. So I just that fueled my passion for it, and wanting to understand more what they're doing, how to apply them, and yeah, how to use them to build soil and soil fertility, and regenerate soil health. So yeah, that's kind of how it all started and piqued my interest. And now I'm just on a journey of continuing to stay up to date with the latest research that's coming out. I think that's really important to especially with soil and microorganisms because new research is coming out all the time. And um, it's so dynamic, it's so complex. It's
Does it contradict itself sometimes? Sorry to interrupt, but does it like,
yeah
will you get like a new
think,
information that contradicts stuff that you thought was already kind of known?
sometimes, but mostly I find that it's like building on itself, and it's like, yes, this is one way that it's happening, but now in addition we found that there's this other way that soil organic matter is being built, or carbon's being sequestered, um, or fertility is being built in the soil. So yeah, we'll talk about some of those new ways today, but yeah, the soil is super complex and dynamic, and we, just want to not get stuck in our one way of thinking about it, and just know that it's always changing. Like the soil is a living being, and it's not going to always be one way. It's not always going to make the same choices. It's always going to adapt to its environmental conditions. So that's why, I think that there's always new stuff coming out as well. So, yeah.
It's interesting because you started you said you've been doing it for 10 years. And so like
Mhm.
Haifa's probably been what, about half of that
Yep.
as like a no-till commercial.
Mhm.
And so when you're in the hydro space, you're more of like a chemist, right? And
Mhm.
then you switch over to no-till and living soils, you become more of a biologist. Um, but you're also playing less, almost like less God. You're trying to understand what's going on, and trying to steer the ship better, but you are not the everlasting lifeblood to the plants with every concoction you give, and if they miss one, it's big problem. Um,
Mhm.
so did you find it easier going, and I say that like I simplify the whole thing, but like once you got it figured out, switching from
Mhm.
hydro to your no-till, have you found greater success kind of like on the upward trajectory? If that makes sense.
Yeah, definitely. I mean, it's it's been, as far as like pests and disease go, like, it's been night and day difference. You still have to do some agronomy, like some soil chemistry, because especially in indoor living soil, like it's a lot different than outdoor soils where you can kind of maintain fertility a lot better, and build soil organic matter kind of easier. But indoors, like we're also kind of limited by, even the indoor lighting spectrums that we have. Microorganisms interact with UV and infrared light, and we don't have those. So it's a little bit harder. I still kind of have to play chemist. Um, but we do the best we can as far as like, um, you know, keeping everything with organic amendments and composting. But I will say, I do think it might be a little bit different. Like I can't just rely on biology all the time.
That's a great point.
Hmm.
That's a really good point because
Hmm.
compartmentalizing yourself away from that connection with nature and
Hmm.
everything that its bounty and everything else, and we've
Hmm.
talked about that with Ross before. It's trying to fill that loop that's closed with something
Hmm.
that's good enough and gonna last. And I'm sure that a lot of a lot of what you do is monitoring the soil's ability to hold those nutrients, and stuff like that. So I think super interesting, and what we're about to see with you, which I think is really awesome, is stuff that a lot of people don't have the luxury of seeing, because you need to have a microscope to do that. And while we pull this up, why don't you like tell us, for someone who doesn't have anything, like what's the best, I guess, what kind of scope could you get to kind of get to this level? Maybe not with a video to share, but like
Mm-hmm.
where would you start? And then let's get into some soil samples.
Yeah, I guess where I started, I like the LW Scientific brand that was recommended through the Soil Food Web School. It's also recommended through like Matt Powers. He uses the LW microscopes as well. So I think it's a really solid brand. They have really good sales and like discounts too that you can find.
And what
If
magnification would you say would be like a good range to be in?
Um, 40x or yeah, 40x magnification to 400x magnification. And then the main thing when you're looking at soil microorganisms, you want to have a shadowing condenser. So when you're looking at microorganisms under a microscope, you're not actually like looking at them, you're looking at a shadow of them because most of them are the same density as water, so you aren't able to see them. So you're looking at their shadow.
And say the name of that again.
Shadowing condenser.
Wow.
Yeah.
Awesome. Yeah, that's gold bar right there. So you don't really have to go Google that one. Great place to start is right there.
Yeah.
I have a question.
Yeah.
Yeah.
So talking about people, what kind of microscopes you get. I don't have a microscope. My wife does, but I'm not ever going to mess with it at all. It's not like I'm going to put my organic stuff into there. So if I don't have a microscope, are you a person, or is the business that we were talking about earlier, do they take samples, like send in samples and you go over them and test them? Is that something your business does or that you do?
Yeah, I do. I recently started doing soil biological analysis for people. I do have a website, organicalexa.com. So yeah, I've gotten some clients, and that's been fun to start doing. Some compost clients, some people that want to improve their compost, some people that want to improve, their hops farm I've gotten. So yeah, it's been cool.
Very cool. So yeah,
That up,
a very valuable resource right here, guys. Like for real, this is the really, it's the second time I've had any of my stuff ever scoped. The first time was Canna Sommelier, I think is how you say that lady's name. Anyway, she couldn't get her microscope to work very well. And it was just kind of difficult because she was brand new. It was all brand new to her too. And I just sent her some stuff, but this is really the first time it's been looked at under a microscope. I've never even got to look at it. I have no idea. I just know it works, but it's really cool that you're doing this. So y'all want your stuff scoped. Now you know where to go.
Yeah, thank you for bringing that up. So yeah, I do a soil biological analysis. So I'll tell you, how many estimated organisms per gram of soil, and then I'll also like take pictures and videos of it for you, which is cool, and do like a quantitative or qualitative analysis as well, and kind of just tell you what I'm seeing and all that. So yeah, check out my website if you're interested. Happy to check out samples for you. But yeah, I'm excited to get into this. Should we?
Yeah, why don't you pull it up and then we can share the screen there. And I think we're going to start with the IMO. Is that correct? This is the IMO 3, Kevin, or IMO 4? I can't remember what you sent to her.
I think
I can't remember either, Harley, but I think it was like maybe Labs, IMO 3 and IMO 4, I think. Did I send you Labs? I don't remember.
No, I don't have Labs, but
okay.
I do have an IMO 3. So we'll look at the IMO 3 first, because personally,
Perfect.
That's the one I'm really most excited about.
Cool.
It's really one of the most biologically active samples I've ever looked at.
Heck yes. You hear
Kev.
that? You hear that, fellas? Are you hearing this? Are you hearing that?
And real quick, Kevin, just for the people that for the people that are joining in here trying to like catch up to speed, because we've been kind of progressive in our episodes of trying to help you guys understand what it is that we're talking about. IMO 1, 2, 3, 4, 5. There's an entire episode of that whole thing. So if you want to get a better understanding, that was the whole point of like do it. We ran through like the first 10 episodes was kind of laying foundational groundwork stuff. So please definitely check that out. But Kev, like the real quick spark notes. Indigenous Microorganisms 3. What exactly is that?
All right. In the KNF world, there's indigenous microbes 1, 2, 3, 4, and 5. This is IMO number 3 or level 3 or stage 3. And that is when you gather up twigs and leaves and a carbon material. And then you also gather up a carbohydrate material, which would be nuts or like grains, wheat bran. Any of that will work, and you combine those two together and you get the right moisture and temperature together, and it takes off. It ignites into just biology all over the place. Basically, what's it used for, you might ask? It's used to inoculate. It is used to inoculate whatever you can inoculate. It's used to mix into soils. It's used to top dress. It's used to make liquid IMO 3, like IMO 3T. You can use it like bokashi. You can put it onto your compost, and it will definitely help start breaking down stuff. I put IMO 3 on my compost and get it real wet, cover it up, and biology starts to show up pretty soon, a few days. I'm sorry, one more
Brian,
thing.
yes,
I think what we said back in some of those earlier.
Live shows. Here we go.
Yep. This is
There
a
we
problem
go.
with mine.
Can
Sorry,
you
we
guys,
lost you, Kev.
Okay, sorry.
Oh,
Go ahead, Lexa.
can you guys see the microbes?
We can see, so if everybody can see, you're talking about the fungal, like the actual like hypha, or are you talking about
Like, am I sharing my screen properly?
Oh yeah, yes.
Yes?
Yes,
Okay.
we see
Okay.
you.
I just wanna
We
make sure
see
you guys can't see my screen right now.
I thought that maybe have been a rhetorical question, but yeah, we do
Oh.
see your screen and there should be microbes there.
Okay, cool. Awesome. Sorry, go ahead.
Kev, what were you? Sorry, you were mid thought there, bud.
Yeah, sorry, I got broke off there. I think in the earlier shows when we were discussing these IMO stages, I said the IMO 3 is the most versatile of them all. It can go so many directions and be used so many ways. So
You also said it's probably the most valuable. Is that correct?
Yeah, that's what I was literally about to say. That one has real value, especially when you're first getting into it.
For this reason.
Definitely has good value, as in it's going to make your soil good quick. And if you like, I make tons of IMO 3. I don't make a lot of IMO 4 and hardly any IMO 5, but you don't even have to make IMO 2 or 1 to make IMO 3. Mother Nature makes IMO 3 out in the woods, but you can make it yourself, and just with carbohydrate and carbon sources.
Alexa, so do you mind kind of explaining, because we'll have like reptile people now on this side of the coin here that are trying to understand this as well. And for people that are familiar with Dr. Laningham, I think that you're actually kind of the new torch or the new wave, Alexa, because you understand KNF as well. When you were saying pigeonholing your thought processes and stuff, I don't see that from you, and I don't see that from Kevin. So I think there's a lot of opportunity here for people to just kind of have fun with this kind of stuff and learn Mother Nature without some kind of predisposed lens to it.
Right, exactly. A lot of, the one-sided, like polarized way of thinking, really just limits you. And, I got stuck in that way of thinking at first too, like, anaerobic organisms or anaerobic is bad and these certain organisms are good, but then when I started like actually making inputs and applying them, I wasn't seeing that happen. I wasn't seeing pathogens come when I see white fungi or highline fungi. They would be called the clear fungi, or problems when there's a lot of ciliates or anything like that. So I think the switch from, pathogens to beneficial, it can be really complex. Like, for example, Fusarium, like a very notorious plant pathogen, what studies have shown is that Fusarium can actually have beneficial relationships with plants under certain like healthy environmental conditions. And it can work similar to like mycorrhizal fungi, where it's like supporting plant growth. And then if conditions become unfavorable, it like switches on these pathogenic genes. And this is like really the same for a lot of different pathogens. um so I think that we need to just look at pathogens in the context of, you know, uh, the environmental conditions of the soil. And, unfortunately, we do like have a lot of toxic environmental conditions. So it's obviously important to be careful and cautious, but it's also important to, understand that uh, there's a the the switch between pathogen and beneficial is a lot more complex, and we can't just like look at a microscope under a microscope and say like this input is gonna be not beneficial for my plants because it's gonna cause, pathogenic conditions. Like, that's what I've seen anyways. Like, always be cautious, I'd say. Especially when you're um playing with new recipes or new inputs. Like, I'm always like diluting things a lot and, being cautious. But in general, like, I've really pushed the limits because I was I want to like cause a pathogen reaction in my plants, like with the inputs that I'm making, and I can't say that I ever have. So, yeah, that's just my 2 cents on it personally. I don't like the fear aspect behind it. But again, just be careful, That's the main advice I have there with the, pathogenic organisms. And, because if we look here, um, sorry, this is opposite of what I'm used to, the way that it shows up on the camera. These types of fungal hyphae, this is classically something that, Elaine teaches that, we should be cautious of because they could be pathogens. And it's this white, or not white, sorry, high line, which is clear. Um, kind of the ace.
And sorry, just real quick. So what she's talking about is that really faint tree
Yeah,
branch looking thing. If
yeah.
you think that we're just looking at kind of like an unzoomed picture here, that's what she's trying to get your focus on, is that right there.
Yeah. Yeah, so that is typically something that, it would be like, oh, watch out for this type of fungi. It is a pathogen, but um, there's I see these in soils all the time, in inputs that I'm making all the time, and it's just I've only seen, beneficial reactions from it, especially in the compost teas that I make. I think a lot of times, like when it's white like this, I believe that it's because the fungi is growing really rapidly. It's has a, food resource and it's growing really rapidly. And then as it ages, it builds more like actually, fungi have melanin in their cell walls, and that's one of the things that makes them brown over time. So as they grow, and carbon as well, and different things. So as they grow and store nutrients, so become different colors. But a lot of, and there are, it's not just bastidiomycetes, which are the um, I guess, most evolved type of fungi that are beneficial. And these are the ones that are, in forests forming mushrooms like you know, boletes and, you know, your classic oyster mushrooms, the ones that we know that are like edible, you know. So those are not the only ones that can be beneficial for your soil. Also, you know, ascomycota can be, you know, beneficial. Some of them form, you know, mushrooms, but
Lisa McCarville asked, "Is that actinobacteria?" I'm not sure. I don't know that word is. Are you familiar with that?
actinobacteria. No, actinobacteria is actually it's filamentous bacteria, and it's actually smaller. So it's in here, you can see it, but here I can like zoom in more. Oh, maybe not. Okay, I didn't like that. Okay, so I won't be able to use this is 100x, by the way.
Yeah, thanks for clarifying that.
100x magnification, so 100 times magnification. So it's not letting me do the 400 magnification. So, but
Alexa, do you mind kind of going through and letting people know what makes Kevin's stuff better? I think a lot of people are looking at this right now more as like art. It looks cool, but they have no idea what's going on right now. And so, do you mind kind of explaining some of the stuff as you scroll by?
Yeah, absolutely. So again, we're looking at these fungi here, and they could be, you know, ascomycota, they could be zygomycota, but just because they're not basidiomycetes, like the ones that are known to be the most beneficial, doesn't mean they aren't beneficial in the soil. So when, and you can see here, a lot of these have like climatospores on them, which are types of spores that they make ascomycota and zygomycota primarily to prevent them from drying out. It's made for survival. So just because it's not like this more evolved type of fungi doesn't mean it's not going to be beneficial in the soil. These fungi actually have better, like less evolved fungi have better carbon use efficiency, so they're able to more efficiently take carbon from whatever material they're eating and make biomass from it, as opposed to just respirating it into the atmosphere as CO2. So this is really important because what builds soil organic matter is microbial necro-mass, which is their dead bodies. And
It's like microbial frass.
yeah. So 50% of soil organic matter is actually just dead microorganisms. So, you know, that's why I'm
Wow,
thinking, yep.
that's
Mhm.
crazy.
So that's why it's like, you know, are any microbes really, you know, bad for the soil? Because anything that you're putting into the soil, like I don't see a lot of times, you know, I hate to say it, but I don't see the microbes like last long term when you're putting them into the soil. I think a lot of it is really short lived, but it's just like fermented food for our gut. You know what I mean? Like when we studies show that when we eat fermented food, like the microbes in that fermented food aren't necessarily colonizing our gut, but that doesn't mean they're not beneficial as they, you know, move through our gut. So I think it's a lot of the same thing applies to the soil. We can think about it as like a fermented food, you know what I mean? So these microorganisms, they go into the soil, and you know, they're gonna most likely not make it that long. Like some will colonize for sure. Some will make it their home long term, but I think a lot of them die because they don't have the food or source that they did in like in your IMO 3, where the food or source is just so abundant. You know what I mean? And so that
What
releases
are
fertility into the soil. So sorry, go ahead.
what are some of these little things that are floating around as you're speaking? There's little bitty things go swimming around every once in a while.
Yeah,
You're able to
so
identify some of those as you see
yep,
them.
those are going to be protozoa. Sorry, I have so much I want to say. I'm just like okay, so there are protozoa, and let's see if I can find any, but the primary ones that I'm seeing in here are Pernonema, and this is a type of euglenozoa. And you know, these are just terms, and it
The big someone asked what's the spiky thing is a nematode. The nematode is those little worm looking things.
yep, so the nematodes are the like microscopic worms, I call them. And I will say these this IMO3 had it has so many nematodes in here. Oh, okay, so this right here that just flew by, that's a clopoda, and that's like a kidney shaped ciliate. And you know, I see these a lot in worm castings.
What are those
There they are.
micro
There they are.
There
what
you
are
go.
those micro movers actually doing? Are they
Okay. Yeah.
eating and breaking down?
Yeah. So protozoa, they
Ooh,
are
a nematode.
primarily eating bacteria, but really whatever fits into their mouth. You know, they can eat organic matter, they can eat other protozoa. Oh, here, there's a
That
Pernonema.
little
Okay, so
that
that's
little
the
black thing.
Pernonema. Um, shoot, I don't know what side it's on because of, but it's kind of small, it's moving. So that's a type of flagellated protozoa. It actually has a really long flagella. Um,
It's that little black thing that looks like an inchworm, right?
no, that's a nematode. That's a small nematode. It was on the other side. Sorry, they're kind of small. They're about 50 to 100. Oh, there's two of them up there. They're about 50 to 100 micrometers in length, and they have a really long flagella that they use to move. Um, yeah, a lot of flagellates that are actually like really hard to see with a microscope because they're only like 5 to 10 micrometers in size. So there are, you know, a lot that are really tiny in here that I do see, but it's probably really hard for you guys to see. And I mean, they're really hard to see just with a microscope in general. It's just how quickly they move is how you kind of distinguish them from bacteria. But uh, flagellates in general, they're super cool. Some of them have two different modes of, or two different morphologies. So when they're moving, they have a flagellated form. So they have, you know, a body, and then they have a flagella, which is just like a long, gosh, how do I describe it? Like a string or something that they use to move. And then when they're eating, they have an amoeboid form, which amoebas have like a squishy pseudopod body. So they use that to um, phagocytize, so take in bacteria through their bodies. So this other thing here is a nematode, the small one there. There's so many nematodes in this sample. It's really amazing.
Is there any way to tell? I mean, so we actually talked about this offline before.
Mhm.
You've scoped predatory nematodes before,
Yeah.
and specifically I'm talking about like the H backs, the oh God, what are the other ones even called? I can't even remember. But the
Mhm.
the other types, Steinernema feltiae and the
Yeah.
Steinernema carbocapsae. Sorry.
Mhm.
Do they look any different? Are they distinguishable, like from a predatory to something like this, or are there different types of nematodes in your experience? Like, is there predatory types? Is there more
Yeah.
detrivore or decomposing types?
Yep. Okay. Yeah, good question. So the nematodes that I'm seeing here are primarily like bacteria feeding. You can tell by like the shape of their mouth and body. And that's how you tell the difference. So there's predatory nematodes, which are gonna feed on other nematodes and other like larval stages
Organisms.
of organisms, like fungus gnats and things like that. And
They're parasitic, right?
yeah, then you have bacteria feeders, and then you have fungal feeders. There's some that feed on like algae and organic matter. There's some that like eat other nematodes. So yeah, there's a lot of different types of nematodes, and you can tell if you can get them to stand still for long enough what their primarily food resource is, or what type they are, if by looking at their mouthpiece. So the ones that I'm seeing here are primarily bacteria feeders. This looks like a nematode, like this long thing here. It might not
Yeah, we
be
see it.
live anymore. Okay, sorry.
Yeah, it was like really long.
Yeah.
And looks like just a dead straight nematode
Yeah.
that
Mhm.
you would find in like a pack of predatory
And to be
nematodes.
fair, this sample is like 2 weeks old, right?
Yeah. Yep. So yeah, like I said, it's really amazing because when I did receive the sample, it was dry and there was still live nematodes in there. Nematodes have a dormant form. It's called cryptobiosis, and they basically just slow down their metabolism enough and like stop moving. So you can tell I have an eye for it because I look at the predatory nematodes we get at the grow all the time, which ones are actually dead and which ones are just in a dormant state. They still look alive. They still have all of their like parts in their body. The ones that are dead look, like they're starting to dry out. Like this one here, I think looks like it's freshly dead. You can kind of see it looks a little wrinkly. I would say that, this one's probably dead. It also looks like, it's maybe starting to get decomposed or something. So,
Back to the biomass, the
the
necro-mass.
necro-mass, yeah. Yeah. So, oh gosh, so much I want to say about the necro-mass. So what is really interesting is that, you know, this bacteria, everything, we have this primary system where we're organisms are the classic soil food web model is taught. We have organic matter, we're putting that on our soil, the organisms are breaking it down, and then that is turning into soil fertility, that's turning into carbon storage. But there are other ways that they have found that this is actually occurring now. And the primary form of organic matter that's stays in the soil long term is called mineral associated organic matter, and it's organic matter that's bound to minerals, and it is resistant to decomposition. So like as soil or as compost like starts to get decomposed, it's starting to form mineral associated organic matter. It goes from particulate organic matter, which is a really easy to digest form of organic matter, what they call labile. And microorganisms, rapidly break that down. And eventually it turns they will break that down. They'll consume it in their bodies. It turns into necro-mass. And then that necro-mass will bind to mineral particles and become mineral associated organic matter. So there's more steps happening to actually building soil, in depths, in the root zone. So a lot of the the study that I found recently is that a lot of the plant litter is not actually becoming stored as soil carbon and soil fertility. It's getting released into the atmosphere through microbial respiration, but through this process where the microbes are eating it and dying, it's actually becoming mineral associated organic matter where it's staying long term in the soil. So,
Hey, let me say this real quick, you guys. What Alexa just went through, I am going to have to go back and watch again. because, but so outside of Leighton Morrison, I believe, I have yet to find anybody who gives a good explanation of what she just mentioned of the, let's see, mineral associated matter, particulate, or ground up associated or ground up particulate stuff like that. I don't even know them all, but I know that there's this new importance to compost and soil textures and things like that. And this is the second time I've heard somebody explain it. And trust me, I'm going out and looking for it. So I'm going to have to hear you say all of that again, Alex, not right now, but I'll go back and listen to it.
No, for sure. Absolutely. So, you know, kind of just to review, because I know that was kind of a lot and I want to make sure I explain it correctly. So particulate organic matter, again, is easily broken down. They call it labile. It typically lasts 1 to 50 years in the soil. And then there's MAOM, or mineral associated organic matter. It's more stable. It's resistant to decomposition. It persists in the soil for 10 to 1,000 years. And yeah, so that's what's like, we're talking about building soil, right? So it's really MAOM that is building long-term soil. Like POM, or particulate organic matter, is great in short term, but if we're thinking of, you know, restoring soil and having these systems work for us longer term, it's mineral associated organic matter. And because they're bound to minerals, it makes them resistant to microbial decomposition. So a lot of times when you see compost that's really broken down, you won't see as many microbes in there because, you know, it's mineral associated organic matter. So they can't, it's not a food resource they can easily access.
So what does mineral associated mean?
It means it's organic matter that's bound to a mineral. And mostly that's going to be, here, let me check my notes really quick. Okay, I didn't write that down, but, clay or metal oxides. That's what it's typically going to be bound to, clay or metal oxides. So, also what's interesting is that there is dissolved organic matter, and this is a precursor to mineral associated organic matter. So dissolved organic matter is plant root exudates, and also it can be formed from just like rain coming down and organic matter, you know, releasing like humic acids and stuff. And then it can also be formed, or dissolved organic matter can come from like compost extracts or compost tea applications, where, you know, you can see the brown humic acid in there. That's dissolved organic matter. You know, when you run water through compost and you make a compost extract and you see this dark liquid come out, that's the dissolved organic matter. And that's you know, quickly taken up by microbes, and then they die, and then they're forming the necro-mass that is binding to the minerals. So
it takes a lot of dead microbiology to create MAOM.
Yeah.
Is that right? Am I right on this?
Yeah, essentially that's correct. So
this is fantastic.
So it's really interesting, and you know, that's why, you know,
these kind of things show you that life is cheap at this level, and so everything is like being consumed and eaten. And I think for a lot of people, like Kevin was saying, they're gonna have to go back and watch this. I don't even know if Alexa realizes the elite mind that she has when it
gold bars.
Yeah. So to break it down, because I know a lot of people are going to have questions and stuff, she's taken basically a decade or more of knowledge, and is, what are we at, like 44 minutes here, and it's kind of giving you all of that. And so we might have to have a part 2 with this. You guys are going to have to understand that it's probably hard for her to break this down because she's at the forefront of this. She's at Camp Compost. She's at the forefront of a lot of this stuff. I think from the cannabis space, everybody kind of already knows that. But from the new world that I'm kind of entering in, and Juice, I saw Juice on there. Mark's obviously in it as well. Is the bioactive reptile space, Alexa, is kind of like the new living soil 10 years ago when people were trying to get people away from the hydroponic setups to a regenerative setup. And it was too complex, I think, at the beginning. It sounded too good to be true. And that's when Dr. Lane Ingham started to break it down. And then for a lot of us here in Colorado, it was the big expos with Jeff Lowenfels and how he would talk about the exudates, or basically allow the plant to communicate with the microbes and say like, hey, tonight I'd like to have a hamburger. Tuesday night I want to have Chinese food. And that communication is how all of these minerals start to actually take hold, especially in a larger raised bed. And these, like, kind of more I'm getting at what Kevin was saying is she's already giving you the plays like you're in the NFL. And so don't get discouraged if you don't know the plays right away, because she's just rattling them off. I don't know if you've ever heard NFL plays, but they're like 45 words long for a reason. And that's how this kind of feels when you first get into it. And so the reptile community that's going to watch this, and I know some of you are actually even watching tonight, we just appreciate you guys kind of dipping your toe into this. And I hope more people won't get frustrated and realize that she's just next level. This is upper echelon stuff.
Thank you for saying that. Sorry. I know I get super nerdy with this, and I just want to share, the most recent research that's coming out, because I guess I don't want to continue to perpetuate, or not perpetuate, but just continue to share this more complex. And I do like to get into the science of it, but when Kevin, he doesn't, have a microscope or know the complexities of these things, but he's still making absolutely incredible inputs and, growing incredible plants with those inputs. So you really don't need to know all this stuff. I just like to get really nerdy, like I said, with it, and do a lot of research, and that's what I do in my free time, you know what I mean? So I hope no one's overwhelmed by that. Like, there's so much more, I could share on that. But I really hope that
you're confirming what Kevin has been teaching for a year and a half.
Yeah,
it's kind of the way I look at it.
Yeah.
I kind of just hope that it opens, people's minds to, like, there's more out there with the soil, and that's my main point with it, is that there's a lot going on, and it is complex and dynamic. And yeah, I don't think we'll ever really truly understand exactly what's going on, because like I said, it's always changing. And I think, yeah, just working with the soil is really what you want to be doing. Like, how do I get back from, sorry.
No, while you're doing that, Juice from Juice's Arthropods made a comment that nematodes and mold is a bad word in the invertebrate world for decades, and around isopods specifically. And I think that's like what Alexa was saying, where there is a distinguishable difference, and there are a lot of different nematodes. Like, that's the one important thing to understand, is that not all nematodes are going to be predatory, and so the word nematode is such a broad statement, right? Like, so if you're worried about nematodes, like especially in a bioactive, maybe stay away from the predatory types that you can buy that might attack other species. Because in my experience, and Alexa, I don't know if you can corroborate this or maybe you can argue, but in like a closed setting, predatory nematodes are indiscriminate killers, right? So like in a lab setting, you put a bunch of organisms, beneficial, harmful, plus predatory nematodes. They're going to go after pretty much anything that has a mouth part that they can parasitize or whatever, parasticize. So from that would probably where the misnomer comes from with nematodes. And when you start to explore the other types of nematodes that fill the voids in the soil food web, which are what's creating the magic beneath your animal's feet in a bioactive setup, you start to shy away from that kind of that misnomer. There's a lot of misinformation out there, not a lot of people talking about the real information. And I'm not saying that a predatory nematode would go after an isopod, because they don't. I'm saying just that probably is where that misnomer came from.
Yeah, awesome. Yeah, I guess I think, you know, I really hope I didn't go over anyone's heads too much. Like I said, I just get really excited about all this stuff and like to share it. And
If you're watching this YouTube show on a Friday night, you're a nerd that wants all the knowledge you can get thrown at you, man. So,
okay,
For real, man.
good.
Especially every Friday night.
For sure. Yeah, no, you know, Kevin, like I really resonate with like you and your story. I struggled with addiction and mental health and
awesome.
you know, a lot of chronic health problems for a really long time in soil and you know, getting back to nature is what really helped me, you know, overcome those things. So, you know, I've heard you share
Wow.
your story before and,
Yeah.
you know, I like to share that as well with me because it's in with Camp Compost as well. That's what we do is, you know, help people get back to nature and it can be so healing. Um, so yeah, I like to
That's
think of
awesome.
compost as, you know, not just from a physical healing context, but also like energetically healing because what is compost? It's the process of death and rebirth. So, you know, when we can apply that to our own lives, what does that look like? It's like, you know, taking whatever worn out behaviors or paradigms or thoughts or things that no longer serve us and you know, transmuting them into something beneficial, composting
Yeah.
them. So I call that energetic composting. And that's, you know, that's what has really helped me transform my life, both physical composting and the process of energetic composting. So that's why I get super nerdy about microorganisms because that basically replaced my addiction.
Yes. What a wonderful story, Alexia. Thank you for sharing that because
Yeah.
you know the humility it takes to be honest and share things like that about yourself are very empowering also
Yeah.
because you open yourself up to flower and blossom. You know, you've spoken out what your evils are and your demons are, and you're recognized, guys. Your demons are recognized. You know, I've told other people about you, you know, and you expose it. And it's empowering to say, man, I was like that. And it's okay to say I was once like that. You know, as a kid,
Mhm.
I was brought up kind of in a strict church. I feel like I still go to church. I call myself a believer and all that good stuff. But you know, it gets oh shoot, I forgot what I was gonna say. Dang it, guys, I'm so sorry. But it got to where it was a little too strict. And it just creates that anxiety that builds up it was like I wanted to be able to say, hey man, I'm just like my parents and I never have problems. My parents had all the problems in the world. I just didn't know it as a young 20-year-old or even maybe a 30-year-old. It's like you got to be able to say, because we don't live in a perfect world, you have to at some point in your life say, I was once like that and I'm not anymore. It's just a sign of progression in life and maturity. So I appreciate you saying that because it's just an example again of somebody who has been blessed, like you said, energetic composting. You just understand that there's a new gratitude in your life because there are things doing work that you're not involved with at all, but you get to reap all the reward and benefit of what they do. And that should be an attitude of gratitude when you realize that microbiology, Mother Nature, God, if you will, has set a system up and it works whether we're around or not. But we can go out and find stuff that Mother Nature is making and bring it back and benefit from it. And we had nothing to do with it. We just gathered up the gift, you know. So thanks, Alexa, for sharing that.
Yeah, that's beautiful for sure. The microorganisms, you know, they're deeply healing. Like, you know, I talk about them as like spiritual beings as well because, you know, what are they doing in compost? You know, they are the ones transitioning it from this waste into fertile soil. You know, so they are, you know, the kind of like angels of the microbiome. They, you know, help things transition from one form to another. You know, they're always supporting like the greater good of the environment. You know, that's what I think anyways. So yeah.
Yeah, dude. Two things again. I always tell people you learn to break open mental loops by closing organic loops. You know, in the mental health world, you loop on something, and that's that unhealthy mental thinking, and it's called the loop. But in the mental world, you want to try to break those loops, and in the organic world, you learn to close them. Like you said, you basically replaced an addiction with an addiction, and it's not like being an addictive person or somebody like that is a bad thing. You've actually recognized who you are, and now you're working with yourself in a positive way. It's okay to get addicted to something that's good and beneficial to you. You know, if you like walking or running. You know, it's just a good thing to replace it with. And I needed something to replace addiction with. I needed something to give me the understanding of joy again, and the ability to appreciate things and to be appreciated and to take care of something and to watch it blossom and bloom. You know, when you have to take care of another thing, you get outside of your own head and you begin to take care of something else and think about other people and their needs. And then you become this healed person once you start to, you know, help other people. When you need help, you begin to help other people. That's how you say it, man. When you need help, you start helping other people.
Yeah.
And I will say, because we glossed over it, but Camp Compost is a perfect testament to that statement right there for Organic Alexa, for Alexa. The event is truly, for me, I consider it my spiritual reset, just because of how immersive it is spiritually. So and the amount the attitude that everybody shares about sharing the gold bars, the good stuff, is so refreshing to see, and such a good reminder of why I am in this community, and like running the show, doing the show, trying to share the information with, you know, no strings attached, and the passions in the soil. And that's really what Camp Compost is. And like, my hat's truly off to you. I couldn't make it this year, which has messed me up. It really has. Like I had multiple friends that were there that I've made from that event going the past 2 years. And anybody that's ever thought about coming to it, make the homage. I would call it like traveling to Mecca, because it really is just an incredible place with an incredible group of people that are there to give you basically everything that they have. So with that, let's get into some questions. We have a bunch of questions.
Yeah,
I know we try
let's
to
do
keep
it.
these shows to an hour. Alexa, if you have to go, we can
No.
probably field these. But we do appreciate your time. We know how valuable it is, and it's Friday night,
No, yeah, I'm good.
10
I can
o'clock.
keep going.
So let's jump into it, Brian.
All right. First question from Rusty Nails. I heard that most of the mold found on IMO collections is just rice mold you could have gotten in the fridge. I think he's also referring to in the early days when we interviewed Luna, she kind of brought this up. And I think that spread as well and talked about these things. So did you want to speak on that, Alexa?
Yeah, I've heard this as well. It's really hard to say like without a genetic test, but I will say that the I guess the IMO2 collections, looking at those under a microscope, it's not, I guess, the diversity that you would maybe expect. So a lot of people say that it's an organism called Rhizopus, which is a zygomycota. It's a less evolved organism or type of fungi. And, you know, it's not to say that it isn't, you know, beneficial or anything, but, you know, I guess I don't have enough experience to say like, yeah, it's always that rice mold that you're collecting, even if you do put it out in the forest. I've tried to make IMO that way a few times. It did not work out. So, maybe, sometimes you are collecting stuff from the forest, and then sometimes it is the mold that's on the rice. So I think I don't have enough experience making IMO. Do you
I mean, I would
think about that?
Yeah, Alexa, here's kind of what I've kind of always looked at it as is if you've got rice and you got something growing on it, I think in a general sense, we can say you're growing IMO on that rice because it's the indigenous microorganisms that's been around and jumped on it. So like it's kind of the Jadam thought also. There is no good, there is no bad. And it's kind of what you were saying earlier. It's a different perspective of looking at things. Like just because it's there doesn't mean it's bad.
Mm-hmm.
And but if you've got stuff growing in a jar that you have IMO2 in, just mix it in. It's IMO growing on your IMO. Just stir it all back in and don't worry about the things growing, because what it is Mother Nature in process of breaking things down and just doing what she does. And just so accept it and know that, okay, this is what is happening, is what is intended and meant and is going to happen. Mother Nature is going to do the job she does. So just let it happen. And you're talking about IMO when IMO2, when you look at it, it's not the microbes you actually want. It's like the introductory microbes you want, kind of. It's like these guys show up to do things so other guys show up to do things, and so the IMO2 isn't the ultimate level. That's why I'm saying that IMO3 is really where you start to see all the development of life take off. And so yeah, you can have isn't rice yeast called koji, or rice yeast? Yeah, is it koji? Is that what you call it? Do y'all know that?
I think that's aspergillus. I'm not
Yeah, that's it. Yeah.
sure about like how it's made, honestly, but
I think it's grown on rice.
Cool.
This question was from earlier from Jim. What keeps swimming by? And I think it was protozoa.
Yeah,
the
protozoa. There's two main types of protozoa I was seeing in there. The genus Clopata, which were those kidney shaped, and then the genus Pernonema, which was the flagellate form. They're flagellated protozoa in the phyla Euglenozoa.
Next question from Rick Easley. I used horse feed mix with my local free mulch, looking good, but the feed has yeast in it. Will the grow faster than my incorporated IMO? I think you meant like, will that grow faster than
Will the yeast grow faster than my incorporated IMO? Yeah.
I'm not sure. Looking good.
It depends what kind of yeast it would be. So in my experience, limited experience, so like if they're adding some sort of like gut bacteria for the horse, it's if it's anything like what's in like a brewer's yeast, or a granular brewer's yeast, or even like a baker's yeast, that yeast is pretty weak. It's not like lactobacillus. But if it's, then again, it would completely depend on what kind of yeast they're adding. But I would say that it's not going to be that aggressive because it's not breaking down the food. Granted, it's dry and everything like that, so if it gets wet, I don't know what would happen. But
I think the majority of the biology found on a lot of IMO1 is yeasts, or are yeasts, if that's the right way to say it. Like, so I think the yeast is totally fine. It's just part of those precursors, or the things that kind of show up first. A lot of yeast does a lot of work, and then other saprophytic microbes move in, I believe is how you say it. Again, I'm talking way over my head trying to name stuff. I just know methods.
No, yeah, I think you're right on. Like it's important. Like that's why I think the IMO methods like work so well is because it kind of like follows succession of microorganisms. So like, you have these, um, you know, uh, I guess primordial like species that kind of show up first and they start cycling nutrients. So like other species can take over. And yeah, I think, I don't know, I think no microbes are necessarily bad. When things do become pathogenic, I think it's probably because of a um of a imbalance. So if you have like way too much of one microbe like taking over in your input or your soil, that maybe might cause a pathogenic condition. It's just like, it's solving a dysbiosis in the system. But, you know, the microbial world is so complex and dynamic. It's really hard to say, you know, what's good and what's bad with it.
Dysbiosis. That's the first time I've heard that word. I'm sure that's opposite of like a symbiosis. Just
Yeah,
meaning not working together.
yeah,
Okay.
exactly. Yeah. So if you have like a overgrowth of one type of organism, nature is gonna always, try to correct itself and kill off, like that's why like monocultures, you know what I mean? They're more prone to pathogens because there's all one type of organism. Like you in a ideal system, you want to have a lot of different types of plants in your system because that's what's going to support the most biodiversity long term in the soils, having lots of different types of plants, and those all support a different microbiome. Just like we all have a different microbiome, all the plants have a different microbiome as well. So I think, environmental conditions, when there's like too much or there's an imbalance or a dysbiosis or too much of one thing, that's when those pathogenic genes start to kick in because microorganisms, they can change their genes, they can change their shape. Like bacteria can um, perform horizontal gene transfer. So they're like always picking up genetic material from their environment and inserting it into into their DNA. So based off whatever is in the environment, they're like changing their function, and that's why it's hard to like identify even like some species in the soil. So, and also soil is like 20 to 40% unclassified genetic material. So it's just like genetic material from things that have died in the soil. So like these organisms have this crazy data bank of of genes that they could, potentially pull from as well. Sorry, that was a tangent.
Oh, great point. Great point, because I mean, we always talk about, more seems to always be better. It seems like it
Mhm.
translates. Talked with Andrew from Benny Mite Biotactics, who he raises predatory mites in the foliage, and even he recommends diversity. When you attack
Mhm.
problems in the foliage, having diversity there translates down below as well. I think it translates in life, but
Yeah.
definitely in the soil.
Yeah, definitely diversity is, essential for like robust, healthy systems. So, yeah, that's why, when you're growing, a crop or anything, you want to have a lot of different plant species in there. I think with been learning about syntropic agroforestry lately. We're doing some stuff with that with Camp Compost next year.
What is that?
It's a type of I guess agriculture where you're growing like trees with different perennials and different annuals all kind of in the same row, and you're mimicking, the layers of a forest. So like, how the forest has like um mm, oak trees, like really tall trees, and then like fruit trees, and then like perennials and shrubs and vines. So you're trying to get as many different plants in there. And the theory behind that is, again, all those plants are making different types of root exudates, and that's going to support all this different microbiology in the soil, and ultimately build diversity in a robust system long term. So it's just like an alternative to monoculture, basically mimicking nature.
Yeah, going back to nature.
Mm-hmm.
Nerd question, and can wait till the end. In my industry, there is a lot of fear of fungal reactions from mitotoxin reaction with arthropods. We talked on this, I think. But from my experience, there appears, and then here's part two, sorry. Arthropods have an immune system. Anything you'd recommend looking out for?
Fungal having a mycotoxin for arthropods. Yeah, I mean, maybe. I guess I don't have any experience with like the, you know, I don't know, maybe this is a question more so for Brian, building those systems. But
Yeah, I think what he's saying is that he's asking that when there's a lot, or he's saying when there's a lot of mold, right?
mm,
That's a big negative in the arthropod world.
okay.
And it kind of like, so it translates to raising springtails as well. And I will say it's a hard question for me to answer, Juice, because the goal with healthy soil and the goal with raising like cultured springtails is a little bit different. But what the observation that I can tell you is that in like a worm bin or a healthy, robust, living soil system that's growing plants, right? I couldn't say definitively with the bioactive, but I could look at Brian and say this guy can show you many different containers where he runs bioactive where there is no mold issue, right? Whereas you translate it over to the homogenized, isolated, singular cultures of springtails, for an example, something that I can speak on, right? So like if you overfeed that system, it will start to grow mold. That mold will start to put off CO2. It'll start to tank out the culture. And it also adds to this that you usually want to be culturing in like an airtight container. So when you have that reaction, I don't know if it's necessarily the mycotoxin or it's the imbalance of CO2 and oxygen in the container, just the excess of CO2 that chokes them out. I guess the point of what I'm trying to say is that when you are fostering healthy, diverse microbiology in the soil, this isn't generally something you have to worry about. And so, like, you have the healthy soil, you can add the layers of springtails. Like, we offer many different colors of springtails that do different things. They're awesome to look at, and they are functional as well. But having that, I think like that foundational, everything else that's both smaller, for lack of even more micro than a springtail, that's going to lend itself like as much, if not much more than anything else in this conversation, really. So the balance and the diversity.
And for people that are newer to this bioactive, my basic breakdown is this. If it's coming from food, especially if you're brand new to this, it's a potential hazard. Especially if you have the vents and all of these things that I hear people are saying with isopods. In my opinion, that's going to open up a lot of possibilities from just an IPM perspective. But from a mold perspective, if things are healthy enough, then the mold that they're probably seeing, and I use that in quotations, is coming from the wood itself. And if it's coming from a wood source, that is a good thing. That is something that you want to have because that is a decomposing food source for isopods. And unless you're doing like dairy cows or something that is so protein hungry, that is the beacon for healthy reproductive. You can add all of the supplements you want, but at the end of the day, the decaying wood, the decaying hard leaves, the ones that actually have the nutrients in them, the magnolia leaves, using the magnolia pods, that's how you're going to get to the upper echelons of building up an immune system within that soil. And when that soil system is that immune system, everything else that's involved in it, Juice, will become alive and thriving. That's where you want to start studying what she was talking about, Dr. Lane Eham, reading books by Jeff Lowenfels. He actually has a third book that talks about mycorrhizal fungi. It's next level. So you could read his first book, Teaming with Microbes, and you would have a nice understanding, kind of like a nice playbook of what is possible within the microbial world. And then you'll start to kind of go down that rabbit hole and understand a lot more of the topics that Alexa was speaking on. But from an isopod standpoint, most of the information that I see on the internet from my experience is completely wrong. And I think it's just echo chambers. Somebody reads it on a Reddit, they say it to somebody else, and then somebody posts it on something else, and then it becomes gospel. And so just like Kevin is kind of the king of this, is just do it yourself. Get the experience. I mean, it's great to listen to a lot of OGs. I've been talking about that for probably a decade now. Learning from others sure speeds up a lot of shit real quick for you. But on the flip side of that is you have to be doing it as well and building experiences and stuff. And I think that's where a lot of this is going to come from. And you nerds that are sitting here with us still on Friday night, if you'll start to actually implement this, I promise you'll believe in it even more. And then that will get you excited, like Alexa was talking about, where now this becomes almost an addiction, or at least a hobby that you want to pursue on a weekly basis. Because I have seen a lot of people kind of come and go with this. They get into it and then they kind of back away. I've gotten into building boxes, putting them in the woods, and failing with every single one of them. And it is discouraging. And I think that's where a lot of people then I'm on to something else, man. This seems too hard. So there's a lot to this. And again, just ride the ride with us. And I promise you that, especially some of the other guests that Mark's lined up, you're going to understand this on a whole nother level. All right, Alexa from Lisa, can she explain how they might morph and how they work in cycle and succession?
Yeah, microorganisms, bacteria can change shape based off of, environmental conditions, like what food's present. There's even protozoa that can change shape based off like if predators are there, they can kind of like puff themselves up and make them look bigger. There are predators around. So this is called pleomorphism, when organisms have like two different shapes. And I think like I was rambling on about earlier, it's like flagellates have a pleomorphism as well. Some of them have an amoeboid form and a flagellate form for moving versus feeding. So they do this, I'm not exactly sure like physiologically how they, how this is happening. With bacteria, they're able to do horizontal gene transfer like really easily. Like it's literally happening like every second, with the bacteria. It's like their language. And so I think that might have something to do with it, with the bacteria. But with like the larger organisms, I'm not exactly sure how they do that, or how they're detecting that. Most of their senses, like they're just single cell, most of them organisms, so most of their senses are coming from like chemicals in the environment. So that's where most of the communication comes from. So maybe they're like detecting, certain chemistry and changing shape based off of that. So yeah, I don't know exactly. That's my best answer, though. I hope I'm pretty close and did that justice. But it's really cool, and maybe we just don't know, because honestly, pleomorphism is like a kind of a new thing that they're starting to look at more. So in understand more, so I think more research will come out about that in the future. How do they work in cycle and succession? So would that maybe that's referring is that referring to the IMO maybe how it works in succession
I'm
or
not sure when this was
how
asked. I guess I
oh
would need
okay
some
she's
reference.
probably referring to like maybe the soil food web. I should have explained that, sorry, did not go over the basics, just like jumped straight into like crazy advanced world. So the soil food web, classic model is you have bacteria and fungi, which are the decomposers, and then you have predators of the bacteria and the fungi, and that's protozoa, nematodes, microarthropods, even like isopods and springtails and organisms like that. So they're eating the bacteria and fungi, protozoa, and then this is how nutrient cycling occurs when they eat those organisms. They release some of the nutrients into the soil. But like I was explaining earlier, they have now built on this model. So we're understanding that their dead bodies are now a part of this. And, there's also, something called the fungal energy channel, which I won't get into explaining, but they're building on how this works in succession. So like I said, the soil is, really dynamic. And, when this Elaine Ingham's like understanding is like the foundation, the basic understanding, and then now the current research is like building on this and saying like, oh hey, also this is happening as well, and in addition to that this is happening, in addition to that this is building soil in this way. So that's what I got really into without explaining the basics, so sorry about that. But
The
So
fungal energy, what?
the fungal energy channel, yeah, that's really cool. I don't know if I should get into explaining it, but
That
this
should be
You don't have to
a
right now. That's
part
just
two.
that's homework. Yeah, that's
I know.
homework for everybody. Go
So
look
I
that
will
up.
give you a podcast though that you can listen to. It's Dr. Christine Jones. If you look her up on YouTube, there's 4 podcasts on Green Cover with Green Cover, I believe, that you can watch, and she explains the fungal energy channel. Yep. And this is just a way that fungi are, building organic matter and storing carbon in the soil through root exudates. So, they're forming these substances like organic acids and glomalin, and their necro-mass and melanin, and that's becoming, long-term storage in the soil. So that's just kind of the basic of it. But Christine Jones, she really gets into the nitty-gritty of it. And so yeah, I guess I'll stop there with that. Oh, I guess I will say, she does kind of say like, oh, the soil food web model that Elaine explains it. She says that it's like not true, which I, don't necessarily agree with. So I don't like those one-sided things where it's like, oh, this is the only way that it's happening, or no, this is the only way that it's happening. But if you look at the actual like research behind all of this, it's like they're both happening. All of this is happening. It's all building soil, and maybe different environments support the soil food web model more than the fungal energy channel model. But in in soils, like these things are all happening, and probably more things that we don't even understand yet, like most definitely. So
and I think the underlying tone too is like the more the more they and then we understand through what these scholarly type scientists are doing, the more you know, the more we realize we don't really know everything, right? So like that's the important undertone here. That's the common denominator is like let's build on this together, you know.
Mhm.
Um and it truly is fascinating and it's all
Mhm.
good information. So
Mhm.
it's too bad one is saying the other is wrong because that's the antithesis of this platform literally we are trying to build onto what we know, which is why it's
Exactly.
such a pleasure to have you on. I think we have a couple more questions, right, Brian?
Yeah, just two more. And to be fair, it seems like in that world, it's like you diss each other in a PhD, so that other people talk about you the same way if you're in like a hip hop world or something. You know, you diss on Dr. Laning and you say all these things so that people say, Dr. Prestigio, what does she know? You know, it does seem like there's drama even if you were selling teacups. There's a lot to this, and I think just again with the experience. I mean, we all can kind of harness this and play around with Mother Nature, and she figured it out a long time ago. So I think us just kind of
Mhm.
peering in here and then saying, "I know everything. This person has no idea what they're talking about," is probably the silliest thing, especially if you're really into this. To
Mhm.
say that you understand the microbial world is a bold, bold statement. All right, from Jeff, or I'm sorry, Jim. Would that be similar to clay and humus compost, the MAOM?
The
I think he's
MAOM. So it's
yeah.
MAOM, MAOM, mineral associated organic matter. Um, I would say yes. So, um, humus compost, that's going to be more so MAOM. Humus is actually different than mineral associated organic matter, though. Humus can become mineral associated organic matter, but it's not. But if you have a in general, from my understanding, anyways, like I said, this is all really new to me as well. So I'm really just starting to understand this and try and break it down. So, from my understanding, is that humus, when you have a lot of like highly broken down material, like humus, like in compost, so like compost starts as like particulate organic matter, like we were saying, the poem. And as the microbes eat at it and die, it's forming humus. And then as the dissolved organic matter in there um, associates with minerals, it becomes mineral associated organic matter. So, I in general, the soil has like all these different things in there. It's just that the mineral associated organic matter is recalcitrant, which means it is resistant to decomposition. So it stays in the soil a very, very long time. Um, and that's, great for building soils, but, it's like, um, maybe in some soil systems you don't want a lot of mineral associated organic matter. I don't know. But yes, in general, I would say if you have a compost that's like highly decomposed, it will most likely have more mineral associated organic matter in it. Especially if there's clay in the compost, because that's one of the things that the um, organic matter can bind to form mineral associated organic matter. So, yeah, I hope I explained that well. But
Uh,
like I said,
Chris from sorry,
go ahead.
I
Go
was
ahead.
gonna say Chris from Country Roots Organic. I'm sure you know him or of him. He always says like it kind of should be his catchphrase, but he's a geologist, right? Like
Mm.
um so he struggles on the but it should be his catchphrase is don't forget the minerals in organics, right?
Sure.
Does that kind of like in a, like if you look at his amendment packages, right? Like whether you go, he's got like and we carry him too, but that's besides the point. There's like a veganic, which is just all like basically minerals and a not a whole lot, like not a ton of NPK value there. but a lot of
Mhm.
minerals that are derived towards either the veg cycle or the bloom cycle. Is that the kind of stuff that you can be adding into your like closed loop system or even outdoors in your garden? But like, is
Mhm.
that the kind of stuff that you can add? And then plus time. It sounds like time is like the big thing for MAOM.
Mhm.
Is that kind of like if you wanted to go buy something today or like do something right now to start working towards
Mm.
that,
Yeah,
like what can you do?
good question. So I think the main thing with mineral associated organic matter is that to understand that dissolved organic matter is the precursor to forming that. So dissolved organic matter again can be root exudates, so literally just the plant exuding the sugars and amino acids. That's dissolved organic matter. The microbes eating that, dying, and then um, their bodies or what's left of them associated with clay in the soil. And then the other way is through compost extracts, actually, has a lot of dissolved organic matter in it because when you make a compost extract, you're running water through it. And what is coming out of that, or you're running, so you have a compost tea bag, you put the compost in there, and you run water through it, kind of massage it because that'll help release the dissolved organic matter and kind of release those things into the water. And your water should start turning like a brown color, and that's showing you, visual, confirmation that there is like this dissolved organic matter in there. And humic and fulvic acids are a form of dissolved organic matter. So that is another thing that you can do to help form mineral associated organic matter in the soil is make compost extracts and put that on your soil. And
That
that
was the pantyhose tech right there back in the day.
yeah, there you go.
Pantyhose on a stick, 5 gallon bucket.
Yeah.
Is it true when you go with like an extract and you emphasize the timeliness of applying the extract, right? So you wash it in the water, you apply it quickly. Is there any merit to maintaining your protozoa and your nematodes and stuff like that in that solution? Like, because I've heard people explain or like express the benefit of doing an extract on top of what you just said,
Mhm.
would be that you're like maintaining, if you emphasize speed, you apply it quickly, you're maintaining your aerobic microbiology better than if you were running a compost tea. Is there any merit to that? Like a
Mm,
bubbled aerated compost tea.
mm. Okay, so kind of a lot to say here because I kind of did stretch the aerobic versus anaerobic thing pretty far in making compost teas because I really wanted to see like, are these anaerobic organisms, if you leave out compost tea for a really long time, like months, is it going to kill your plants? And that's not, what I've seen. But I will say that yeah, your biology, is going to change over that period of time. So I've um experimented with making, compost extracts versus aerated compost teas, and then also static compost teas as well, because a lot of people don't have like access, or when they're starting out don't have a compost tea brewer. And so I started being like, I wonder if there's like an easier way to make a compost tea. Just, kind of let compost, sit out in water for a few days. And um, I will say that making an aerated compost tea is, where I'm seeing you're going to get the most organisms to grow, like especially fungi. And You can get good protozoa growth as well, like the most diversity in general you're going to get with the aerated compost tea. But if you want to make a static compost tea, let's say you don't have like access to a brewer, like that's definitely something you can do. And I started making protozoan teas too. So a good way to do that is just kind of let the compost tea sit static for, 5 days or so, and the protozoa are eventually going to take over in that tea. Um, and, you know, those protozoan teas are really only, I would use them like with caution, because if you brew a bunch of protozoa in your tea, like I said, a lot of these are not gonna stay in your system very long. They're gonna die and like release their contents in the soil, and a lot of times, or you have more potential to burn your plants because you're releasing a lot of um, you know potentially available nutrients and things into the soil that could burn your plants. And I have burned plants that way before. But you can also use protozoan tea like strategically, and they'll help decompose like organic matter, so you can put them on a compost, or before you plant, put that on your soil, and it'll help speed up decomposition while they're in there, increase fertility. Again, you're going to get a lot of that necro-mass in there. And so basically, yeah, I don't know if I answered your question, but
Yeah, the answer to the question is it's like bigger. It's bigger than the question.
okay.
No, and it's a lot to think about, and like
Sure.
there's not a lot of people that have scoped that kind of experiment
Mhm.
and can
Yeah.
talk on it. So it should give a lot of people a lot of hope that it's kind of hard to screw this up.
I
And
tried. Like
yeah,
I've really tried.
yeah. So like you don't always believe what you read, and maybe just keep your mind and horizons a little bit
Mhm.
broader, because you just heard it from Alexa. I mean, there's people that would say if it's sat for 48 hours, or it's been bubbled for 56 hours, or 60 hours or whatever, god forbid, it's no longer any good. And
Mhm.
essentially what you're saying is like that's not necessarily wrong, that's not necessarily true, it's not necessarily not true, depends on the
Mhm.
environment, a
Mhm.
big time obviously.
Mhm.
But don't always assume that that's wrong. Just
Mhm.
proceed with
Oh, no.
caution and dilute it like Kevin does.
Yeah.
Just dilute it down. Like
Yeah.
Yeah.
don't
Yeah.
stress it.
I will say I was helping an old man have a grow. I left with my wife on vacation for a week. I came back and he hadn't fed the plants the compost tea. It smelled like hell. I got pissed at him, to be honest. I was like, dude, you were probably the laziest grower I've ever seen in my life. I guess because of that, he then fed them to the plants, killed everything. So
Huh.
I think there's
Yeah.
something to, like,
No,
if you're
I
just
mean,
lazy, like just lazy upon lazy upon lazy, then you're probably going to get what you deserve when you would
yeah,
speed up the microbial life and then make it go anaerobic like that. I mean, when
for
it
sure.
smells to high heaven, that's usually a good reason for you not to proceed.
For sure. I mean, there have been people who've, told me like, oh, I have killed a bunch of plants or gotten diseases from compost teas. Like, I just, I don't know. I've watered in like straight JLF that smells horrible. You know what I mean? That sat there for months and didn't dilute it at
That's
all, and
that's
it did not.
all Kevin uses.
So it
Exclusively
really.
all what Kevin uses, by
Yeah.
the way.
Use it today.
He probably
Yeah,
smells like it right now.
You probably do.
not like that stuff. Like, I think it's great, like mad respect, but oh my gosh, the smell is terrible, especially like in an
His wife
indoor grow.
loves
I
him.
got banned for making those, because if you open that lid in an indoor place, it smells like that for the next 3 days. It's
No
so
doubt,
bad.
man.
But I've let like compost seeds sit out for a month, and watered them in, and I've gotten good plant reactions from it. And But people, that's why it's like always be cautious. Just always just test what you're doing and dilute when you're trying out a new recipe. That's really the way to go. And the whole aerobic versus anaerobic thing, maybe has some validity in some contexts, but like the soil, you need to have both anaerobic and aerobic for it to function properly. Like for nitrogen fixation to occur, you like anaerobic environments are preferred because the enzyme used for nitrogen fixation is really sensitive to oxygen. So it prefers these what's called microaerobic environments, which is a lower oxygen, not necessarily anaerobic completely, but a very low oxygen environment. In addition, there's the soil redox reactions as well, which you need a balance of both anaerobic and aerobic conditions to have proper soil oxidation and reduction reactions. So that's the how the soil is accepting electrons, and that affects your nutrient availability. So that's why, again, with the one-sided thing, it's not aerobic or anaerobic, it's both. You want both.
This last one is a personal question, and I'm glad Kevin came on because it's kind of fun. It's for me, it's my wife and I love to debate, and I feel like this book gives it some merit on both sides. So I wanted to bring this up. Can you guys see that right here? Can you see that screen?
Yep. Now we can.
Great. All right. So Darwin's Black Box. It's a pretty interesting read.
Yeah. I've read it.
Michael B. Okay. So the bacterial flagellum, that when he brings up that, again, if you haven't read Behe's work, this is a very interesting book. The Galapagos Islands, he breaks down a lot of this stuff. It's I don't know. I would almost use the word, it's like hidden from the general public. But anyway, in the book, he talks about a bacterial flagellum, which I will pull up here. Y'all can see that now. Does it take a sec?
We just see Darwin's black box. You might have to change which screen is sharing. This is the problem with doing live shows, baby.
Yeah, let me do that.
We'll cut that out.
Yeah, right. We don't edit shit.
Ain't no edit nothing. You get
all
what
right,
you
can
get.
you see that? All right, so
Yes.
Here's the bacterial flagellum. And Michael Behe, I wanted to kind of give it to the panel here. He's talking about that there's no way that this could have ever evolved, right? There's no real adaptation or anything. This is basically like an onboard motor that would be spinning at, like, let's say 10,000 RPMs. Stops on a dime and can spin 10,000 RPMs in the other direction, like instantly. And so I just wanted to bring this up since we were playing around with the microbial world and this. I have fun with religion as well as on a scientific side of things. It intrigues me more than anything else in life. And this one book, I think, is very eye-opening. Again, when it comes from the Galapagos Islands, most of the belief systems that I think a lot of people have come from finch birds. And so I think there's a lot more to learn from my microbial world, and the bacterial flagellum is one of them. I kind of wanted to again throw it over to Kevin, then I was going to throw it to Alexa, but since you've read the book, I mean, what are your thoughts on what I was talking about?
Well, what he does in there is just try to give evidence for instead of what, like, well, he deals with intelligent design and some other things that Darwin, he kind of refers back to Darwin, but it's been probably 15 years since I read this book. However, I do remember this part of the book when he talks about that flagellum and the tail and how, God, I can't even remember. Dude, I can skip through all this and tell you that, yeah, I think there's a deeper method and design to what we see in nature. There's, like I always say, it's Mother Nature's way of doing things, meaning it happens with you or without you. In 1 million years from now, it's going to be happening. 1 million years ago, it was happening. We just live in this short little blip of a bigger system that goes on and functions properly. It functions the way it's intended to. It can overcome. It doesn't seem as though it's perfect because it has ups and downs, earthquakes and fires and things like that. But it finds its way. It continues to live because it has that basic element of I live and I survive and I bring life to this place. So I think there's a deeper meaning. There's definitely an intelligent design to things. I think in the codes of DNA and RNA, this thing, they're just so amazing how they're put together and what their intents are and what they can do. There's so many available options in the DNA code, and it's amazing that we're not it's amazing that we're not always just awkward and weird, messed up when we're born. Just the amazing ability that everything went right for us to be alive and do what we're doing. I have to give credit to something else than besides my good genetics. I want to give credit to something else. I want to be thankful, and I want to say, man, I want to be thankful for the life I have right now, the peace of mind I have, the family that I have. I want to be thankful to something for that. I don't want to be like, yeah, I did it. Because I'm not the ultimate end. The point is, I would never say, look at me, I'm the ultimate end and you'll find happiness if you look at me. I have to point to something else because I don't have that for anybody. I have to say, look to something bigger than me and bigger than you to find that joy and peace. And I think that there is something bigger than all of us, and that something has participated in this creation, and it works the way he or she intends it to work.
Do you have anything? I mean, I'm not asking for your religious beliefs, Alexa, or anything, but do you have any kind of viewpoint on this? Had you ever heard of bacterial flagellum before that? And the reason why it sticks out is because, at least Behe's point is, there's no way that it would ever have adapted. It either was created or it would have never been there, right? And so it's kind of like science version that there is some kind of intelligent design, and other people say God, universe, or whatever verbiage you kind of want to use. But I love it because it's on such a small scale of things, and like quantum physics. And if you're really talking about all the kind of weird woo-woo stuff, this is kind of at the forefront of it for me, is this book and this little species.
That's cool. Yeah, I definitely am interested in reading this book. I guess, yeah, I do have, some thoughts. So I always think about life as like fractal microbiomes. So you can think of like humans, like we have a microbiome, right? But you can think of humans and animals and as the microbiome of like the earth, or what some people call like Gaia. And then you have the earth in the solar system, that's the microbiome to like the galaxy. And then it expands further and further, probably in larger directions, but then it also expands in like smaller directions as well. So like our microbiome, that has a microbiome, like the virome, right? And then the viruses probably have a microbiome, and it just keeps getting smaller. So all of creation is kind of like reflected on itself in a fractal form. So like you see similarities in the way that things happen from the large scale of thinking the galaxy to all the way down to like the small scale with how the microorganisms. So I guess that's my initial thought, just seeing this. But yeah, I definitely like to read this book, and I like thinking about these concepts as well.
Mark, you have anything, man? I just wanted to end on kind of a fun note, since you guys are upper echelon with the microbial world, and I don't know, just having fun on a Friday.
Man, I don't. I abstain, but I love the conversation, and just the thought of it. It kind of goes to show you that like nature is the guide, and the stuff that we don't understand is probably the most important part of the whole thing. What we actually do understand is just for novelty at this juncture. When you're talking about nature, granted, and I say that with a grain of salt because Alexa is a perfect example of how it's not just being used for novelty, because you are actually applying things that you're learning from the soil that you guys have in your indoor beds at Grown, and steering your inputs in a way, and maybe even steering the concoctions of microbiology that you're putting in. Sounds like you kind of are mad scientist over there. And sounds like some great stuff to follow up on part two, because
Yeah.
I would love to keep talking for 2 more hours. We love to try to keep this to an hour, but I had a
Yeah, you
feeling
said that.
this was going to go over, and this always happens when we have bright people that are passionate about what they're talking about. So,
Yep.
really appreciate your time on this show. And before we sign off, let's run through shout outs, and we'll start with Kevin over there.
You bet. You go to okcalyxshop.com. I got that IMO3 she was showing on sale. It's fire. The stuff I made that like I finished it, and like the next day I sent it off to you. So that was definitely some good stuff. But okcalyxshop.com got lots of stuff up there. I'm soon going to have my new films that I did last fall. We're getting them all grown out this summer. I'm about 3 weeks away from my outdoor harvest, so it's getting close. And then October 19th, I'm having the autumn immersive class all day long. It's me and Growcast putting it together. I'm teaching you how to go out into the woods, how to find stuff, process it, bring it back, and how to use it. And you all will take home all the stuff that we make. And I have a lot of inputs that I've already made, I'm going to make, and then we'll make a bunch out there. So go to growcastpodcast.com and you can sign up for that class. I believe we got 10 tickets left. How about you there, Brian?
Yeah, it's a big weekend for us. I actually had to rush home. I don't know if before you guys got on here, I was smoking and out of breath because I wasn't sure I was going to make it. But we have a huge expo courtesy of the All-American All-American Reptile and Plant Expo at Castle Rock. This is a show that has like class to it, and it's hard to be a vendor at the show and all that. And so it's kicking off this weekend. My wife and my kids are there as I'm speaking to you guys. So really excited to be, even be allowed to vend it at a show like this. There's some national brands that are there. And so just kind of continuing to climb the ladder. And I'm appreciative of the Sloan Reptile crew and Robin from Redline Shipping, kind of just putting together a class act. Mark, what you got, man?
Just gratitude on my side. mibeneficials.com. You can buy our stuff there. And we're going to have some sweatshirts coming down the pipeline here pretty quickly, so keep an eye out for those. But gratitude for Alexa spending 2 hours with us. And also, of course, for Brian and Kevin spending 2 hours here talking about this stuff. And you can't hide passion.
Pleasure.
Yeah, you truly can't hide passion, and I think everybody here, Alexa absolutely included, just exudes passion. And those are the type of people
No
that you
doubt.
can really learn a lot from, because they're the type of people that will truly give it all to you. And that's why I want to shout out Alexa's website and Alexa personally, but what she's a part of as well, I think, is profound, and you're kind of like an embodiment of the community, the regenerative community here in Michigan, which I think is a great example for everybody, all communities around the country and the world, and it all kind of starts at Camp Compost, because I just like the mission is so pure, and there's no ulterior motive, and it's not really about selling. It's about giving and sharing. And so it's in a very special place, and it's crazy because this is by far the longest I've sat with you, this 2 hours. But I've been to that for 2 years, and Alexa, I can assure you, Alexa is not the reason why I came to Camp Compost, or like even anybody that I didn't even get a chance to see you because you're busy running around doing everything. But everybody at Camp Compost is just as passionate. I mean, it's just like, that's the spiritual reset I'm talking about, is like, you can actually go have these conversations with just about anybody out there and talk about something that gets everybody's fire lit. And just knowing that there's others out there like this. It's not just like hearing it through the earpiece on the drive to work, from somebody like-minded. It's like meeting these people is truly incredible. So long-winded thank you, Alexa. And
Thank you, Alexa. It was
but-
fantastic.
Yeah.
real quick before we sign off, how do we, other than that right there, organicalexa.com, best ways to find you, and how can we support you?
Yeah, that's it. First of all, thank you so much, and thank you so much for, having me on. I'm sorry if I just kind of didn't start with the basics. I probably should have started just like with the basics, but I just get so excited and ramble and get nerdy and yeah, so maybe next time we can start with the basics and move up from there. But if if you want a sample compost or soil sample that you'd like me to do analysis on, go to my website organicalexa.com, and there is a contact box, so you can just send me a message on there, or you can send me a message on Instagram too. That's totally fine as well. So yeah, I just appreciate this opportunity so much to do the live microscopy. It was so much fun for me. The IMO 3 sample was absolutely incredible. So, definitely I would be ordering that stuff. I might order that stuff. So, yeah, I mean, I could seriously keep talking for all night probably, but yeah, just really appreciate the opportunity and you guys listening to me, and yeah, I hope I explained everything as well, and I hope that yeah, like I said, I'm learning a lot of this stuff too, so like what I say today could totally be like not true tomorrow. That's how fast our science is moving, so just take everything with that in mind. So anyways, thank you so much, I appreciate it.
Thank you, everybody, for tuning in. Until next week, keep your horizons broad.
Oh yeah, sorry. And
Yeah.
campcompost.com, and campcompost on Instagram. Yes, super incredible event. Really just an incredible community that we have that comes up, and yeah, you'll have an amazing experience, and we love to have see the community grow. Yeah.
Let's grow together, boys and girls.
Let's go.
Have a great week and weekend, everyone.
See you guys.